Today I will talk about the neurosphere assay, and in order to do so, I will give you just briefly an overview about the background. Okay, so we have a really different student type, let's say from medicine to neuroscience, from bachelor, master, PhD students, so different backgrounds. So I think it is good and important to first know a bit about the background. So regarding neuroplasticity, it's the ability of neural circuits to be remodeled as a result of different environment or activity. So the ability to delete or create synapses. So can you see my mouse walking here?
Yes.
Okay,
Go ahead. Good. Everything is okay.
That's good. Okay, so we delete and create synapses. And this is important for learning and memory. So nowadays, we also know that generation of new neurons in the adulthood, so the adult neurogenesis, is also important. So the immutable brain, adult brain, it was a dogma for almost 100 years. And this started with Santiago Ramon y Cajal. So probably from the people from Spain, so you all know them. From the people that are not from Spain, you should also know his works. So Santiago Ramon y Cajal is considered the father of neurobiology, and he did this amazing drawing. So this is a hippocampus, a part of the brain. And he did these amazing drawings with these microscopes. You can see these microscopes, which are nothing like the ones that we have nowadays. And so he said something that was, so more than 100 years ago, which was in the adult centers, the nerve paths are something fixed and it's immutable. Everything may die, nothing may be regenerated. So at that time, he thought that it was impossible to have some kind of regeneration in the brain because of these really complex structures and this complex network. But he also said something really interesting, which is it is for the science of the future to change, if possible, this harsh decree. So at that time, so the idea was that this was not possible. So new neurons were not, so they are post-mitotic cells, so they were not capable of dividing. And he thought that there was not possibility to have new neurons in the adulthood. And this was because of the techniques that he had at that time. But of course, as a pioneer, he said, maybe the science can change. And I want to guide you a bit about how science has changed along these years. So in the late 60s and early 70s, there was these first papers showing that neurogenesis could occur in the adulthood. And these first papers were in rodents. And then in the 80s, there was this pioneer paper showing that neurogenesis could occur in the songbirds. So every spring when they had to learn new musics, that they would need neurogenesis for this learning. However, in the 80s, there was this publication that really considered that neurogenesis was only limited to lower, let's say, this lower evolutionary order animals, such as rodents and birds. And so, again, these studies were a bit stopped for some years. And so only in the 90s, I used to call it rediscovery, there were these two seminal papers showing that really neurogenesis occur in adult brains in two specific regions. So the ones that we call neurogenic niches. And so these two neurogenic niches, they have these neurostem cells. So what are neurostem cells? They are stem cells that have, so they, as the other stem cells, have capability of proliferate, of self-renewal, and they are multipotent. So this means that they can give rise to neurons, astrocytes, and oligodendrocytes. So this was quite something new and important at that time to realize that in adult brain, we have neurostem cells that really can give rise specifically neurons. And so what are the neurogenic niches? So they are localized in two areas, one in the hippocampus and one, so this is a sagittal slide of rat brain. And you can see here the hippocampus. It's a very important region in the brain. And here it's the lateral ventricle. And so the other neurogenic niche is localized here. And so this hippocampal neurogenesis is localized in the subgranular zone of the dentate gyrus in the hippocampus, and this subventricular zone is here in the lateral ventricles. And to give you just a bit more of detail, so this is a cornel section of a brain, and you see here the lateral ventricles, and if you zoom in, you'll see that these neurostem cells are these blue cells, so the type B cells. So these stem cells in the SVZ, they are very interesting because they can connect to the, so they cross this epithelial layer, ependymal layer, sorry, and they connect to the lumen of the ventricle. So they receive connections from the CSF, from the cerebrospinal fluid, and they also have connections with the blood. So these cells are quiescent, so they are really slowly dividing, but they can, when activated, they originate these precursor cells that already divide more fast. So that these red cells, the type A cells. And then they originate the type C cells, which are the the A cells, sorry, the neuroblasts. These neuroblasts are the immature neurons. They migrate along the rostral migratory stream, and they are incorporated in the olfactory bulb. And they are integrated here as interneurons. So summing up, so we have the type B cells, which are stem cells, very slowly dividing, give rise to the precursor cells that then give rise to neuroblasts that then migrate and integrated as mature neurons in the olfactory bulb. And I already say here that they are important for our memory and discrimination. So this was shown with rodents, that when the discrimination task is very easy, so there's two smells and the animals, so the smells are very different, so there is no need of the SVZ neurogenesis. However, when there is a task that is very, very difficult, the neurogenesis from the subventricular zone has an important role in this other discrimination. Regarding the hippocampal neurogenesis, in this case, we also have this, the names are different. They are called type 1, type 2, and type 3, but the structure is more very similar, in which the type 1 cells are called neurostem cells. They are also quiescent, slowly dividing, that activating give rise to the precursors that are already dividing faster. Then they give rise to the neuroblasts, these type 3 cells, that then migrate, but they migrate very short distance. So they are incorporated only here in the dentate gyrus granular layer, okay? And by being incorporating here, they are important for learning and memory, okay? And particularly the most accepted function of adult neurogenesis is its role in pattern separation. What does this mean? This means that two memories that are very similar, the neurogenesis in the hippocampus is important to discriminate between these two memories. This hypothesis states that if there is no neurogenesis, there will be, of course, as I said, neuroplasticity, but there will be an increased interference, okay? And if there is new neurons, so new neurons being born, the interference will be lower, and so it will be more easily this discrimination, so this pattern separation. So in the hippocampus, it's very important for learning and memory. Although also nowadays we know that hippocampal neurogenesis is also important for mood regulation. So this was really to give you an overview of the importance of neurogenesis, and now adult neurogenesis, and now how can we study in the lab. So this braining, when we started or when we submitted, the idea was to have experts that could explain you some important methodology that we do it in the lab, and also methodology that we can use to reduce the number of animals to be used, okay? So we use these models in vitro to find new modulators of neurogenesis. So in the lab, my group is interested in finding new modulators of adult neurogenesis. And in order to do so, one of the first things that we do is to start by studying it in vitro, okay? Before going in vivo, we start doing this in vitro. So we use this neurosphere assay. Okay, I will explain briefly, and then I will show you a video protocol that we published 4 years ago. So usually use rat or mice, so they are postnatal animals from P1 and P3. We can use also adult animals, although the efficiency of these will be lower. This means that we'll still have neurospheres, but the neurospheres will be, we will have less neurospheres, and they will be smaller, okay? Because the capabilities of the proliferation of these stem cells in the rodents will be decreasing along the aging. Okay, so after sacrificing these pups, we remove the brain, and then we remove the areas that we are interested, so the SVZ or the dentate gyrus in the hippocampus. Then we culture these SVZ or dentate gyrus cells under proliferative conditions. So this means that we give growth factors, then I will explain you a bit more about this, which are the EGF and FGF, and these cells, so we remove the tissue, but these cells, the stem cells, in vitro stem precursor cells, they are responsive to these EGF and FGF. So only these cells that are responsive to these EGF and FGF will grow, will proliferate and grow as neurospheres, which are clusters of neurostem cells. Okay, after these, we plate the cells, and I will also explain you briefly later how we plate it. We plate and we leave it without the growth factors, and then we can treat these cells with the different drugs that we want to test. So after some period of days, it will depend on what you want to analyze, these cells will start to migrate out of the neurospheres, and they will start to differentiate. So this we can see whether our drugs that we are testing are inducing neurogenesis or not, okay? So I want you to give you, so you saw the, I hope you could follow the video, and I'm sorry for all these issues, but I hope you could follow and to have an, I think it was, my idea was to bring the video that you could follow all the protocol, okay? Because just showing you slides with a timeline, it's not informative enough. So I wanted to show you some important steps. So one is the cell dissociation. So as you heard, so we sacrifice the animals and we remove the tissue, and then we have to dissociate this tissue into individual cells. So we use trypsin EDTA. So trypsin is a protease that is involved in the breakdown of proteins. So it will break down these proteins into shorter peptides. So in our protocol, this trypsin is derived from porcine pancreas. And this is trypsin EDTA. So EDTA is a chelating agent, so it neutralizes calcium and magnesium. This will increase the enzymatic activity of the trypsin, okay? So this is important for this dissociation. Then, so we dissociate, and then we grow these neurospheres, so these cells in 3D clusters in these neurospheres. You can see here the images. For that, we need to have these growth factors, which are EGF and FGF2. So as I said before, these cells, these neurostem cells, they are responsive to EGF and FGF. So they have these receptors, EGFR and FGFR1. So that's when EGF and FGF is present in the medium, it will bind to these receptors. It will activate a cascade, a signaling pathway that will allow this proliferation of the stem cells. So this is how you see this neurosphere. So it comes from one cell that by being activated, this EGFR receptor and the FGFR receptor 1, when activating by making this signaling pathway, it will divide and dividing more and more cells, creating these clusters in 3D clusters. And how can we then differentiate the cells? So to differentiate the cells, what we do, so we have these neurospheres and we need to plate them. So we have this cover slip. So after having these neurospheres as 3D, so these cells are very highly proliferative. So we cannot study differentiation. At this stage, we are only having proliferation. So we need to plate these cells in a cover slip. So this usually it's a glass cover slip, and if we put the neurospheres, it will not adhere. So to make these neurospheres capable of adhering to the cover slip, we cover the cover slips with a substrate. So for the dentate gyrus, we need to use PDL and laminin, and for SVZ, we just need PDL. So these are both substrates. So PDL is a synthetic compound that provides a positive charge in the cover slip. So when it's possible, the cells which are negatively charged will adhere more easily. And the laminin is an extracellular matrix that plays a crucial role in this adhesion and migration. So it's very important, particularly for dentate gyrus cells, for these cells to really adhere to the cover slip. And then we remove the EGF and FGF. So by removing these growth factors and by having a substrate, these cells will start to migrate. And you can see here, this is a pseudomonolayer of cells. Why do we call it pseudomonolayer? Because we still have, so when we plate the neurosphere, we still have some layers, let's say, of cells in the neurosphere, but then as they migrate, they start to have this monolayer of cells. So I think it's also important to show you, so you saw in the video some images at the end that were fluorescence images, right? So how do we do this? So we take advantage of some cell markers. So some cells have specific markers which express antigens that we can take advantage and then use an antibody to recognize them. So we can know if these cells, for example, if they are stem cells, they will express these markers, which are SOX2 or nestin. If they are immature neurons, they will express these markers, which is double cortin. So we take advantage of the fact that different cell types expressed different antigens that we can use. Then we also saw that in the video that we evaluate the proliferation. So you saw that dentate gyros have not lower, but smaller neurospheres, and the SVC are bigger neurospheres. So we can evaluate proliferation by adding a thymidine analog, which is BRDU or EDU. So they are both thymidine analogs. So when there is a proliferation, so during mitosis, this BRDU or EDU is being incorporated instead of thymidine. So then we can analyze and find these by also during the staining. So how do we do this staining? So this is called immunocytochemistry. So we have the plate where we have the cover slips, and so we have the antigen. So the antigen is what we want to observe. So we use a primary antibody that recognizes this antigen. So for example, the double cortin for neurons. Then we use a secondary antigen antibody that is connected with a fluorophore. So then when we go to the microscope, we can see this color because it was the secondary antibody that had this fluorophore, okay? So this is very simple explanation of immunocytochemistry, but briefly what we do. So we remove the cells, we fix the cells. We can also, if you have questions, I can explain you how we fix the cells. So the fixation is to maintain the cells intact as they were alive. Then we incubate with the primary antibodies, we wash, we incubate the secondary antibodies, and then we also use a dye for the nuclei so that we can stain all cell types. And then at the end, we can go to the microscope. And you can see here, you saw this in the video, you can see here the difference between a neurosphere from the subventricular zone and from the dentate gyros. And here already when we plate and they start to differentiate. And here also, it was also in the video, you can see that these cells, neurostem cells are really multipotent. So here it's markers of neurostem cells. This is already immature neurons, and this is OPC, so oligodendrocytes precursor cells. So you can see here DIV2 means the in vitro 2, so that's why it's more immature cells, and the in vitro 7, it's mature cells. You can see astrocytes, you can see mature oligodendrocytes, and also mature neurons. So we can really follow these cells from being neurostem cells until they are differentiated cells. And I want to give you briefly an example of how we can study this at the lab. So I have a published work. It is already some years ago that we published. And we were really interested in studying the endocannabinoid system. So we probably know that cannabis is the most consumed illicit drug. So in some countries it's licit, but in most it's an illicit drugs. It activates, we have endogenous cannabinoids, and it activates mainly two types of receptors, CB1 and CB2 receptors that are widely expressed in the brain. And these are the two main endocannabinoids. They are very interesting because they are synthesized on demand from the postsynaptic side. They are released mainly to the presynaptic and activate these presynaptic receptors, and then they modulate the release of neurotransmitters. So we know that cannabinoids regulate different functions, feeding behavior, pain, cognition. And what at that time we wanted to see was whether the activation of the cannabinoid receptors was also modulating neurogenesis. So the first, one of the first things that we study, we did this in the subventricular zone. We also did in the hippocampal data gyros. And you can see here an image in which we have in blue the nuclei. As I said, we always stay in the nuclei. And in green, the BRDU positive cells, so the cells that were proliferating at that time. And we counted, so this is a CB1 receptor agonist, and we counted the effect that this agonist had in proliferation. We observed that CB1 activation promotes proliferation. You can see here an increase. So we have more green cells. And what was really interesting was that blocking CB1 or CB2 was inhibiting this effect. So we, at that time, we were very surprised. We thought that using, so as you know, probably know, agonist is activating the receptor, antagonist is blocking the receptor. So when we were activating, we have an increase of proliferation, and we are blocking both CB1 or CB2, we lost this effect. Then we also saw study neuronal differentiation. So we look at double cortin positive cells. So here it's in green. So we looked at immature neurons, and we saw that activating the cannabinoid receptors increased neuronal differentiation. We also look at neuronal differentiation of mature neurons. So we look so we gave a pulse of BRDU, so it was incorporating during proliferation. And at the end, we saw that these cells that incorporated BRDU were mature neurons. So we saw that these agonists were again inducing neuronal differentiation. And we also analyzed neuronal maturation. So also in the video, we said that we can measure the dendritrogenesis. So we can analyze with these cultures, we can analyze the length and also the ramifications. And we saw that the agonists increase the neural length when activated, and also the branch points, so also the ramification, okay? And so the study is very long, but we showed both in SVZ and the dentate gyros that there was a crosstalk between CB1 and CB2 that modulate neurogenesis. And since we have this cross-antagonist phenomena in which we blocked one receptor and we lost the effect of the other receptor, we saw that probably there was heteromers occurring between the CB1 and CB2 receptor. Okay, so this was an example, a really short example of what we can do in the lab, but you can study different drugs. So as I said in the beginning, imagine that now we have an idea and you want to study this new drug, whether it was inducing proliferation, differentiation. It can be inducing differentiation of neurons. I focus today on neurogenesis, but we can study differentiation of astrocytes, of oligodendrocytes, okay? Also in the video, we said that we can expand this culture. So this is very important to limit or reduce the number of animals. So we can, by starting with the reduced number of animals, we can expand this culture. So this is with the passages, okay? So it grows off in neurospheres, then we dissociate, we grow again in neurospheres, we dissociate. So we can grow different, we can passage and have more, increasing the yield of stem cells. And then, as I said, we can study survival, proliferation, and differentiation. And really, the neurosphere assay has this is a powerful method to mimic physiological condition, okay? Because it grows really as a 3D structure. And how can we apply this? So we can study the stem cell properties. It contributes to drug discovery, to understand disease pathology, and also to develop therapeutic strategies. And today, I focus in this neurostem cells, but we can use this neurosphere assay, so the sphere assay in different, not only in the brain, but from other regions in the body. As limitation, it is enabled to trace quiescent cells. So what does it mean? I said that really the stem cells are so the type 1 cells in the hippocampus, they are quiescent, so they divide very slowly. And in the neurosphere assay, but also in the other monolayer of cells, it's very difficult to trace these because they are not so responsive to the EGF and FGF. And also the neurosphere may aggregate, and at the end you can have so many neurospheres all together, all aggregated, that you cannot have this monolayer of cells. And also they are highly dependent of culturing conditions. So we have to be very careful when performing these cultures. Finally, I work. And last but not least, I want to ask you to follow us on social media. So if you scan here, you can go to our different pages, LinkedIn, Instagram, and X. And yeah, that's it for now.